Hello and welcome to the Kamla Show, where we bring you interviews and conversations with technologists, entrepreneurs, filmmakers, and other news from in and around the San Francisco Bay Area. And today we are going to be talking about science, drug research, and medicine with Dr. Sara Kenkare-Mitra. She is the Senior Vice President, Development Sciences at Genentech. Sara was recently elected to the Institute of Medicine, and she is one among the 150 top University of San Francisco, California alumni, and UCSF is one of the top medical schools in the country, right?
Yes, that's right.
Okay, so congratulations on all your accomplishments. It's been 18 years at Genentech?
Almost in October.
18
yes,
years.
18 years.
Okay, so Genentech itself is a very different kind of a company. It's a research-driven company founded by a scientist and a venture capitalist.
That's right.
And the DNA of Genentech is very different for another reason, because you did something called recombinant DNA. What is it?
Recombinant DNA.
What does that mean?
So recombinant, as the word suggests, is combining different DNA pieces to create a DNA that's usually not available in nature. So that way you can actually create DNA based on the need. So it's a technique that's used either in the laboratory but also to make drugs. And in fact, now there are a number of drugs, there are proteins that you can make from recombinant DNA, which are then grown in cells to produce large quantities of proteins or antibodies that we use as products.
So that is different from the chemically produced drugs?
This is very different from chemically produced drugs, because chemically synthesized, most of the drugs we take, aspirin, Tylenol, things that people are used to having to treat challenges, are synthesized using chemistry and that organic synthesis. And so this is distinct from that because it's more biological and it's more consistent with the biological systems in terms of producing a drug.
So the word is biologics?
It's biologics.
Okay.
So small molecules are molecules that are produced via chemistry, and those are small because their molecular weights are around 150. It's a technical term, but it's a small molecule. And biologics, something like an antibody, is actually biologically produced, but they're also large molecules. So their molecular weight is many, many thousands of times greater than the small molecule.
So now that we've got the 101 of Genentech out, what exactly do you do? Because as head of developmental sciences, what does that mean? Are you in charge of new drugs?
Yeah, so in development sciences is a kind of what we call a translational organization. When I say translational, we kind of sit right in the middle, working very closely with our colleagues that do the original discoveries on targets that we want to target to actually create a drug, as well as our colleagues that are clinicians that are developing clinical trials to translate those scientific discoveries and help them discover new drugs and then put those through clinical testing and the science behind that to understand diagnostics, like that is what patients should we target, what type of changes that are happening in the patients allow us to make some decisions around what doses we should give patients, how much should you treat, how long should you treat for, who should you treat. All of those scientific decisions that happen is based on the work that we do during the drug development process, and that's what happens in development science.
So that's what, so you're sitting in the middle of these two groups.
That's right.
In a sense. And do you then help the drug, do you bring it to market because of the work that you do with these two groups?
That's right. So we work right from the beginning of the discovery phases of a molecule, take it through the entire process, and are involved in actually working with the FDA in the final, making the approval of the drug or bringing it into the market. So we span the entire gamut from discovery all the way to putting a drug in the market.
Okay. So you have been involved with 5 or 6 of them. I cannot pronounce the names, so I'm going to leave it to you to pronounce. But 2 of them are for eye.
Yes.
One, I think, a couple for oncology, cancer.
Yes.
And the latest one in 2013 was something that I think you were recognized for that, right?
Yes. So essentially, the Genentech as a company works on a number of different areas. One is oncology, which is cancer, and that's where our largest drugs that are made at Genentech are made in oncology. But we also have drugs in autoimmune diseases like, asthma. We're working on other areas like multiple sclerosis, etc. We also have drugs for the eye, and one of them is Lucentis, which is used to treat something called age-related macular degeneration, which really can help people improve their eyesight, particularly the elderly who get this disease. And we also work in other areas like neuroscience, but we don't have any drugs yet in that area. So oncology, clearly, I've worked on a number of drugs. The company itself has delivered drugs like Rituxan, Herceptin for breast cancer, Tarceva, which is in fact not biologic, so it's a small molecule in that.
Oh, so it's a
chemically synthesized drug. So we do chemically synthesized drugs, although initially that's how Genentech grew as a biologics company. But today we have the gamut of all kinds of molecules. So I've had the good fortune to work on a number of these compounds, which are drugs today. Xolair for asthma, and then more recently Kadcyla, which is a drug that I was personally involved in, not just from my organization's perspective, in the discovery of that drug. And it's used today, it's in the market for the treatment of metastatic, which is breast cancer that's spread beyond to other organs.
How did you get involved in this last drug that you discovered? Or
yeah, so the Kadcyla.
How many years did it take, first of all? How many years?
Kadcyla, I remember working on it around 2001 to 2003, and it was the way I got involved. Clearly, everything is a team sport. Working in this industry is a team effort. Is that I was involved with some of the researchers at that time to really figure out ways in which we can come up with this molecule, which was the concept was very interesting. It's a whole new concept, not new now, but very new at the time, which is called antibody drug conjugates. And the idea is, in a very simple ways, is if you take an antibody and it carries like a payload, and that payload is something that gives it that extra punch. So in this case, what is Kadcyla?
So that antibody acts as like a Trojan horse.
The antibody acts almost like a Trojan horse in a way. The antibody goes and binds to the tumor cells because it's meant, it's been created in a fact to be able to bind to certain receptors on the surface of those cells. In this case, it was the HER2 receptor, which is found in about 25% of the women that have breast cancer. And so when it binds to the cell, typically it's an unarmed antibody, but when it has the payload on it and it delivers inside the cell, it actually delivers the payload in addition. So it fires the payload, and the idea is it kills the tumor. In addition to its own capacity to kill the cell, it adds that extra payload which kills the tumor. And the other advantage was that the hope is that one day such a therapy will completely, or idea like this, the concept like this will completely relieve women of taking chemotherapy, hopefully.
Oh, so this medication you don't have to take chemotherapy?
Well, at this point, women usually have had Herceptin and taxanes before they get onto Kadcyla. But while they're taking it by itself, even though it's carrying that payload, which is like a chemo payload, it doesn't cause the side effects that women get when they get chemotherapy. So the idea is very elegant, but it's very hard to create, and we were successful in creating that antibody drug conjugate, and I'm just very proud that it's on the market today, and it's showing a lot of promise and changing women's lives. But the idea and the concept obviously is one that we would love to use for other situations in cancer.
So you found a new delivery mechanism.
A new delivery mechanism.
And that is what is very interesting about medicine and pharmacology and drug research today. It's these new ways of delivering medication.
That's right.
It's not the old ways.
That's right. It's there's one concept which is actually coming up with new medicines, because you learn about a new target and then you come up with new medicines. But we also always want new technologies, new ways to deliver, new ways to tackle a problem, and innovation is clearly key in the whole industry.
And since we're here in Silicon Valley and Genentech is in South San Francisco, how do you work with other tech companies in the Bay Area? I'm thinking of 23andMe, only because that is the company that I think, has quite a large database of
That's right.
Gene pool database.
That's
Is that
right.
the right word?
That's right. It's a genomic database. So, as many of some of the public viewers would know, Genentech recently did a deal with 23andMe, and we also did a deal with another company called HLI, which is Human Longevity Inc. And in both these deals, we have trying to get access to data from human samples. Now, in the case of 23andMe, because of the work 23andMe has done in the past, they have a large cohort of patients with Parkinson's disease. And this allows us access to about 3,000 patients with Parkinson's disease whose genomes will be sequenced as part of the collaboration.
And how does it help you?
Yeah, the way it helps us is that when you sequence the genomes, there's a whole technique called whole genome sequencing analysis. The analytical technique allows you, hopefully, to find clues to the disease, and clues for us as a company, targets, areas that we can then target and come
What
up
are
with
these
new
clues?
drugs.
What are these clues?
These clues might be genetic variants, some variation that's happened or a mutation, for example, that exists in those patients that have Parkinson's versus normal, healthy people.
So these mutations are your answer.
The mutations are a clue, because we go, okay, so there's a mutation there that might be something involved in the disease process. It may not be the origin, but it may have a role to play, and that allows us to go back and then study it further. And if we find good results, we could then start targeting it. We could say, okay, the science allows us to understand, what is it, what is the mutation, what is the receptor that we want to target, and find ways to actually target it. And that's how we create drugs.
So that brings me to a very natural question of this personalized medicine and medicine that can be specifically targeted to patients. How is Genentech working on that? Is that something that would be of interest to you? I mean, to me it seems like.
Sure. In fact, I think the biggest premise of Genentech, and Roche for that matter, Roche is the company that owns Genentech, is around personalized medicine. And what we mean by that is is the whole concept that not all drugs are meant for all people. That drugs, that there are certain patients or certain parts of the patient population that probably will have that drug would have the best effect on, and we want to know what those patients are. We don't want to just bombard the entire population with a drug if it's not meant to do that. Now, some drugs maybe they work in everybody, and so this is part of our drug development process. Throughout the process, we are looking for what we call biomarkers that allow us to determine if the drug is showing a greater effect in a certain patient population within all of the patients that we are looking at. And once we know that, then that could become the basis by which we would then plan our final trials and develop a drug in that population. So if I could use an example of a drug that has been on the market for a very long time is Herceptin. And Herceptin is used to treat women with breast cancer, but it's only used to treat women with breast cancer that are HER2 positive.
What is HER2 positive?
So HER2 is a protein, let's say, that's present on the surface of tumor cells, but it's not present on all breast cancers. So when a woman goes
so some have the HER2, some
have
don't.
it, some don't. So about 25% of the female population with breast cancer has HER2. So when a woman goes in and gets a biopsy of the breast tumor, it's usually typed. And HER2, for example, is one example where if you've got HER2 positive breast cancer, then Herceptin will become a part of your regimen. And that's terrific, because before Herceptin ever came on the market, this type of breast cancer had the worst prognosis for a woman. But today, because of the presence of Herceptin, and we've now got a number of other treatments for HER2 positive breast cancer, like Perjeta, Kadcyla that I just mentioned, because of all these drugs, HER2-positive breast cancer is actually a very treatable, if not almost curable, disease.
And that's because you found the biomarkers.
Well, that's because we were able to identify this. We actually made the antibody for the HER2-positive patient. But how do you do that actively during the development phases? One, you know all the biology of the target, and so you have some clues. And then in clinical, when you're developing the drug in the clinic, you're all looking to see, are those patients that actually have that biomarker, are they the ones that are responding better than everybody else? And so that requires a lot of statistical and data analysis. But if at the end of the day we see that, you know, we started with a hypothesis that it's HER2, and then we look for the HER2. This is true for every drug. We have a few hypotheses, and we're looking for those specific things. But if we actually do find statistically that, wow, that patient group has a much increased effect from this drug, then that could be a patient population we would go in. So that's exactly what's personalized medicine, because we have now personalized that medicine for that patient population.
What is your day like?
My day, you know, at one time when I was in the lab, obviously my day was very much in the lab kind of a scientist. Today, obviously, as a leader of an organization that's very science-based and R&D based, I spend a significant part of my day around scientific decision making. And what I mean by that is projects. We have got a huge number of projects
I was just thinking, you must have.
Yes. And so, you know, I spend a lot of time either in reviewing some of that data, helping to make those decisions as to whether, you know, firstly technically whether the analysis that's been done sounds reasonable or not. What steps should the team take? Because every investment that's made, the next stage, for example, if you go from phase 1 to phase 2, is a significant investment. We want to make sure that the data is robust enough and is giving us, you know, giving us enough data to move forward. And the idea, you know, the mantra, so to speak, in our industries, fail fast, fail early.
So it sounds like the tech industry.
Yeah. So, you know, we want to do everything to see if the drug, you know, if there is any loopholes, if there are things that it can't be a drug. We want to know that early. But if the team shows a robust, you know, decision making around that, we feel that the drug actually or the molecule does have the characteristics, we want to take the risk to move it forward. So a lot of my time is spent with that. Another huge part of my time is obviously spent around people, which is by running, you know, to run an organization like that, I do have to make sure that I, number one, spend time hiring and developing the right people under me directly.
So you're personally in charge of hiring decisions?
Yes, because I have an organization of about 500 people. And so at least my next levels, you know, I'm very involved in managing and making sure that I've got the right people and they have what it takes, you know, they have all the resources and help from me in terms of making sure that, and they're very, the people that I've had hired in my organization, that are on my team are just fantastic. And so they then have the big job of actually running their organizations, etc. But I also spend some time on just thinking about the culture of the organization and making sure that we continue to remain innovative, that we continue to remain a fun place to be. And because the life cycle of a drug is very, very long, it takes a long time to go from a discovery to make something a drug. Usually in the around 12 to 14 years, let's say.
Has technology helped in reducing that time?
There has been a lot of efforts, and we are constantly making efforts to find ways to, to become more and more efficient, so that the actual time that it takes to actually develop a drug is shorter. But because of that, there are no fast rewards in terms of the work you do, and this requires a level of, resilience, long-term thinking, not getting disappointed or getting disappointed, but really.
So how do you deal with your disappointments? Because you're constantly looking for drugs, and you're looking to see if you're in the right path.
That's right.
And there are going to be disappointments because some of your you may have a hypothesis that may not be proven statistically.
That's right.
How do you deal with that?
That's right. In fact, more often than not, we are in that boat than the moving forward boat, right? Because because of the sheer numbers, there's a lot of failures and there's a lot of things that don't work based on your hypothesis. But
so how do you help your troop to get up and go?
Well, part of it is our training. You know, it's the nature of the work we do. We've been always taught to run experiments that allow you to, you know, negate your hypothesis, so to speak. So part of it is the training, and I think that does help. And I speak for a lot of the people in my organization that are used to this idea. But in addition to that, it's challenging. You know, we try and make sure we help people see how the patient really matters. And to me, at least for me, that's very inspiring. I'm very inspired by the fact that the drugs that we develop are really making a difference in someone's lives. We try very hard to keep the patient and the science always on the forefront, you know, whether it's on a decision making, whether it's in the day-to-day, and that's where the leader's challenge lies, is to do it in a way that is still authentic but continues to be inspiring for the people.
What are the skills you had to learn as you were climbing up the corporate ladder? Because you started off as a researcher.
That's right.
And then, you know, you worked in a group and now you're head of a group.
That's right.
What are some of the skills you had to learn, and is it different for women?
Okay. You know, so let me first then go to the skills in general, and then maybe we'll come back to the topic of women. I think that, you know, the skills as a scientist, when I started out as a scientist, clearly most of my skills were around focusing on the science. And one of the things I always advise people that core competency continues, particularly if you want to be in R&D. Even if you are a vice president or a director or at any level, your core competency really matters. So you never want to really go away from that, because that's the strength you bring to the table.
That's your DNA.
That's the DNA. And then how that fits into the broader process, and really knowing that is important. I think in addition to that, the skills that I believe are critical are ability to really influence, the ability to communicate well, because communication, not for the sake of communicating, but really to enable good decisions, making decisions.
Did you have to take classes on how to communicate well?
I've had some training, but
no, I'm not saying that you should, but
yeah.
but that's one of the things that when you were climbing up the corporate ladder, these classes
yes.
that you have access
yes,
to.
Yes. So I've definitely benefited from, you know, coaching, etc., which I think, you know, you should always use every resource possible, because as you're growing up the ladder, and that's where the issue of women comes in too. We do find that women, although this is true of men too, but women sometimes are more hesitant in public situations or in group situations to really speak up, and it doesn't come naturally. It could also be cultural, and for me, I think it was a combination of both, and I had to really overcome that. There almost came a turning point where, you know, as the groups started getting bigger, the stakes were bigger, and you had something to say, but you were just hesitant. That was when I really, you know, had to think through, come up with tools and ways to really, you know, let the guard down and really be able to participate in the discussion and the decision making. And that was key, and I say that as an important flex point for a lot of people.
You almost didn't join Genentech because you have a special needs child.
Yes.
And you almost gave up your career.
Yes.
What made you come back?
Yes, so you know, when I had my daughter, who obviously has special needs, at the time I had to take time off because I was trying to deal with what was on my plate and helping her through her challenges. I had, because of being a person in science, as I was doing that, obviously her helping her was the primary thing on my mind. I wasn't thinking of anything else. But I always heard from everybody, you know, once you're out of science, you basically cannot come back in. And that was ringing in the back of my head all the time. I was very fortunate at the time, to have a few mentors, particularly in the academic side. I came from UCSF. My graduate advisor, who still continues to be a very rich and important part of my life, was...
What's his name?
His name is Dr. Les Bennett at UCSF, and he's always been a great advisor, and someone, not advisor just for the science, but just for life too. And just having him and other mentors in my life who said, just focus on this, think about that later was very helpful. The 2 or 3 years that I had to take off, I was completely zoned in on what I had to do to help my child. And then it was just by sheer chance that I actually got a call from Genentech. And I almost didn't take the job because I wasn't sure if I could balance, what was going to be a lifelong situation with a career back in the sciences. And I had to give myself some timelines that said, well, if I can't do it in X amount of time, then I will quit. I was very fortunate, I believe, to work at Genentech because, number one, I think the culture was it was that DNA matched my DNA, and that was very helpful.
So there is a recombinant.
There was a recombination of some sort. And then secondly, Genentech, and I think this is true of most industries in the Bay Area now, clearly wasn't so focused on, what time do you get in and what time do you leave. And that helped me a lot to maintain that balance, which I needed even more at that time because my daughter was still very young and I was still trying to figure out, how to make everything work. So in some senses, it was sheer serendipity that I got back, but it was the kind of the shoulders of the giants or the mentors that I have that helped me to build that confidence that said, keep going, and that's what I did.
And your daughter now is happy with the way you work and take care of her?
So. I think so. I think she's happy and I hope she's proud.
Okay. What do you do for fun?
Because
you're such a research-oriented person.
Well, I love to dance, so I put on the music and dance, and then I love reading books. So those are the two big things that I do that allow me to relax and enjoy myself.
And you don't take vacations?
I do not. I don't like travel vacations. So I do take vacations, but they're usually very local based and very calming to the senses.
Okay, so we are lucky because you're on vacation and you're able to come and do this interview.
Yes, that's
We've been
true.
talking for, I don't know, over a year.
That's true. It took a vacation to actually do this.
It took a vacation. Sarah, thank you so much for stopping by and talking to us. It was fantastic. And before I say bye to you, where do you see yourself in the next 5 years? Or 2020, where do you see yourself?
That's a terrific question. I don't know exactly what I would do in the next 5 years, but I really do want to take everything that I've learned and use it to influence the industry, whether it's the biotech industry, but overall the healthcare industry. And I'm very interested in being a key influencer in this area. I don't know what that exact role will take, but I know that that's the direction I'm headed.
You're headed in the right direction. Healthcare is on everybody's mind today.
Yeah.
So we wish you all the best and thank you for stopping by.
Thank you, Kamala.
And thank you for watching. We'll be back again next week with another show. And if you missed any of our shows, we are available on YouTube. All the interviews are on our YouTube channel. And we'll be back next week, like I mentioned. Thank you for tuning in.